Pajamas! Alright, so you probably tell I'm a little tired because this is like my gajillion thing I'm doing today. But today we're going to talk about atypical chromosome structure. We've already started talking about regular chromosome structure in one of our previous videos. So now we're going to talk about what happens when the structure gets a little bit... stupid glares. When the structure gets a little bit wrong. There we go. All right, so. All right, what can I do? Problem solving. Yeah. Beating the glare. Yes. All right, so there we go. No more glare. Just gotta shove all my stuff back so that we can see the board. All right. So, as we were saying, chromosomes. The typical structure of a chromosome is as follows. We have and right there, that is the centromere. And then we have the, what we typically call the short arms and the long arms. Now, if you're wondering, Miss Komar, they look kind of equal. Why do we call them short and long arms? Well, very glad you asked. There's a very good answer for that. Generally, because a lot of times, the centromere can either be close to the center or close to one of the edges, or specifically close to the top. So we might have something like that. Got our centromere, short arms, long arms. So that's pretty much just why we call it what we do. Now, in terms of atypical structure, as you guys know, the prefix A means, not normal. So atypical structure of a chromosome means that there is something not normal about it. And we can actually divide these up into different groups or kind of, types. So we can have a duplication. Yeah. So let's say in our duplication, this is our chromosome. All right. So maybe what happens is during replication or during something called translocation, this piece gets taken out. Well, now that means that this chromosome is now missing that information. So now it's actually missing a complete section of it. And depending on how much of it was taken, could be one gene, could be multiple genes, could be portions of genes. So any number of ways this could be affected. So that would be an example of a deletion. Duplication is, well, what if instead of losing that piece, that section during DNA replication, it got replicated twice, and we end up with two of it. Well, again, it's now longer than it should be, so when it goes through protein synthesis, question is, how is that going to be read? Is it going to be read like the normal gene? Is it going to be twice as expressed? Or is it because of the duplication, is it going to be read at all? Maybe it won't be. So that's what we can talk about in terms of deletions and duplications. And those can occur any number of ways. Could occur during DNA replication. It could occur during something called translocation. Now, translocation means that something is moved from one thing to another, and not necessarily in a good way. There are two main types of translocations that we're going to talk about. So we have our first one is called Robertsonian translocations. So we have purple is one particular chromosome, and red is going to be a completely different one. They're non-homologous. You can tell that by the size. So in Robertsonian, what can happen is a piece of this one can break off and just kind of attach itself to another. So this one ends up missing information. This one ends up gaining that information. Again, it's not supposed to be there. So how does it get read? Does it get read at all? Does it not get read? Is it going to affect how the other genes on that chromosome get expressed? So that's what we call Robertsonian translocation. This one ends up too short. It is missing information. This one has information that it shouldn't have. Remember, these are not homologous. Speaking about, non-homologous, the other type is called a reciprocal translocation. Now, reciprocal kind of means it goes both ways. Now, in the Robertsonian, what we saw was that it was just a one-way transfer from the purple to the red. But in reciprocal, think of it like crossing over, but not between homologous chromosomes, between non-homologous chromosomes. So what could happen is the bottom bit of each one could be lost, and they exchange it with the other. Again, those sections that were exchanged do not have the same information. They do not have the same genes. The thing about crossing over, it works because you're just exchanging different versions of the exact same genes. These don't have the same information. It'd be like if I said, I went up to a friend, and in the example of crossing over, which occurs between homologous chromosomes, I exchange with her one book, and she gives me another book in return. They're both books, but they're just different books. In reciprocal translocation, it'd be like if I gave her a book and she gave me a pencil. I might feel a little cheated. She might feel like she won the lottery. But, it's complete difference in terms of the information that is being shared. The next thing we have is something called an inversion. And when we talk about things being inverted, we kind of mean that they've been flipped around. So let's see. Let's say we have, I'm going to put this side now. A, B, C, D, E, F, G, H, I, J, K. So in an inversion, what happens is a piece of it actually gets flipped around. So we can have, instead of what it should be, we have A, B, C, D. We have E, J, I, H, G, F, K. So what happens is during replication, or again, maybe it was a incorrect translocation, this section was flipped backwards. So now again, the genes are all out of order, so that could affect when they get expressed, if they get expressed, any number of things. So these chromosomal mutations do tend to have larger, more severe effects than just regular gene mutations, because it's going to affect multiple genes as opposed to a gene mutation which only affects one gene. Let's see. We also have isochromosomes. So what happens in an isochromosome? Now, normally, during division, let's say meiosis 2, during anaphase 2, I draw us two different colors just to make a point. Now, how this should split in meiosis 2 is we should have like that. So each one has the short arm, centromere portion, and the long arm. But in an isochromosome, what happens is instead of splitting this way as it should, what if it splits perpendicular to that? So let's see. So for whatever reason, maybe the spindle fiber is attached to it incorrectly, and instead of splitting down the middle like that, it instead splits this way. And what you're left with is two short arms together and the two long arms together. Like that. Well, that means that they're going to miss whatever genes and information are on the missing arm, and they're actually going to have a full duplicate set of the genes that are on the same arm. So isochromosomes is when, instead of splitting properly, the centromere gets split perpendicular to that, and instead of getting short and long, so all the genes on that chromosome, it ends up with two copies of either the short arm or two copies of the long arm. All right, and then there's one more chromosomal mutation I want to talk about, and that is something called ring chromosomes. Now, what those are is exactly what it sounds like. So we have our chromosome. And as we said already, in a previous video, during replication, we actually lose a portion of the ends. Now, the ends are called telomeres, and they shorten every time we go through DNA replication. So what they produce when they first separate are what's called sticky ends. And what this is a ring chromosome could be at those two sticky ends. Ended up moving around and fusing together. So now that they've moved around and formed this ring, let's say A, B, C, D, E, F, G, H, I, A, B, C, D, E, F, G, H, I, obviously not to scale. What happens is if this separates again, let's say for the next round of DNA replication, it may not separate where they first connected. So it could end up separating somewhere else. And we could end up with, maybe it separates this time between E and F. So we could have E, D, C, B, A, I, H, G, F. So now we see it as all kinds of crazy. Again, a lot of times these different types of disorders, they tend to be, or these chromosomal mutations do tend to be pretty rare because they have such big effects that oftentimes the individual will not survive, simply because our cells don't know how to read a lot of these chromosomal mutations. So I hope that helps you understand chromosomal mutations. Hope everyone's staying healthy and well. Again, if you have questions, hit me up in our Google Classroom or by email. And just until next time, stay well, stay healthy, guys. All right. Bye-bye.